Okay, so we are recording. It's when you want, Arnaud.
Okay, so hello everybody. Thanks for joining this fourth Let's Talk About Ambrosia lectures, and I thank Zoe and Nicolas for accepting to make a short lecture on the topic of Ophraella. So the idea is to have a short presentation and then question and answers, so an exchange about the talk. It's recorded, so it will be available for those who cannot attend. And I think I said everything. So Zoe and Nicolas, the floor is yours.
Thank you. I will share my screen. Okay. Is it working?
Yes.
In a big. Okay. Great. So I don't know if Nicolas is here, but I will start introducing, and if he wants to say something, he will join. So thank you for inviting us to talk today. Our work is about Ophraella Communa, which could be a biological control agent for Ambrosia artemisifolia in France, so common ragweed. It's a project that was launched by Nicolas and Vincent Le Sieur, Nicolas DESNEUX and Vincent Le Sieur, in 2021, I think, and we are funded by Adopt IPM, which is a Europe-China joint action to increase the development and adoption of integrated pest management tools. And we are also funded by two regional health agencies, INRae PACA and Auvergne-Rhône-Alpes. So that's it for the introduction. I will present briefly what is Ophraella Communa. It's beetle from the Chrysomelidae family. For those who don't know, you have here some pictures. So it's a beetle native to North America, just like common ragweed, and his primary host is also common ragweed. You have to the right the cycle, so it will overwinter as an adult during autumn and winter, and in the spring the adult will emerge, start laying eggs for the females. About 6 days later you will have the larva, and about 12 days later the pupa, and then around 7 days later the new adult. So we can say that is around 1 month to have a new generation, one full cycle. And so in France we estimate that we could have 3 to 4 generations per year. And when you reach the 3 to 4 generations at the end of the seasons, the population density are high enough to have an impact on the production of pollen and seeds for the ragweed. So it's interesting. To have an example, here you have some pictures. So in the lab, to the left, you have the plant before we put Ophraella Communa, and then after. So you can see that it's only the stem left, so it's really effective. And you have also an example from Italy, where you have in the field a ragweed plant that was completely defoliated by Ophraella Communa, and all the plants were not touched by the insects. So it's really interesting because it's really specific to Ambrosia artemisifolia. So we could use Ophraella Communa as a biological control agent in France to complement existing control methods, and also to reach areas that are difficult to access, like riverbanks or urban wasteland, and also to target areas that we don't know that are under colonization by ragweed. So the first plant that will arrive will already be targeted by Ophraella Communa. That's our goal. Here I will just present the program launched by Nicolas. Okay, I will present for him. So it's a 4-step project that was launched in 2021 by Nicolas DESNEUX and Vincent Le Sieur. The first step was about the importation of the beetle in the laboratory containment. It was done with insects imported from Italy to the Cicero facility in Montpellier. And then the second step was to test the host specificity in order to evaluate the risk and to do the application file to ask for the introduction of Ophraella Communa in France at the ANSES, so the French organization for introducing new species in France. It was accepted by ANSES recently, but it's now in the validation process by the ministry. So we are still waiting. It was done in November 2023, and we are still waiting, unfortunately. Also, then the third step, it's concerns the mass production of the beetle and the establishment of release plant. So we target this for 2024. We are currently doing this, preparing for mass production. And we did that in collaboration with Chinese Academy of Agricultural Sciences, because they already do biological control of ragweed with Ophraella Communa, and so they have this since 2008. So it's a long experience, we can say. So they are helping us for that. And so the final step is about the deployment of Ophraella Communa, so release of the insect in the field. And we plan to do this in 2025. About the two first steps, so it was already done. It was part of my master's studies in 2023. So I will just present here my results that I had and that we published last year, so in November 2024. We did both non-choice test and choice test, but here I will only present the non-choice. So what is non-choice test is that we put insects, so either adults or larvae, in a cage with non-target plant species. So all the species are here. And we just waited 3 weeks and look if they will lay eggs, if they will eat the plant, if they will survive, etc. So here you have above you have the viable eggs that were laid by the female. To the left, it's Ambrosia artemisifolia, so the common ragweed. It was our control, and all the other non-target plant species. And the same for larval survival rate, because we test also larvae. What we saw was that there is a low risk for indigenous plants. So all the plants are to the right, from Bidens cernua to C. solitarialis. I forgot the name, sorry. All these plants are native to France, and they are indigenous, so we didn't want them to be targeted. And we can see that there is no eggs laid, except for Carpes cernium, but it was really low. And all the larvae died. So there is no risk for them. And what we can say is that they are all phylogenetically distant from ragweed. So even if we didn't test all the plants, we can say that all the plants that are also distant, genetically speaking, we also be bad hosts. So it's really interesting. So and it's consistent with data from Italy and Europe. So we were quite happy about that. Concerning the non-indigenous plants, so sunflower and also Elianthus tuberosus and all the plants that we tested, there are some eggs laid and some survival, but it's quite low compared to Ambrosia artemisifolia. And so there is still a strong preference for common ragweed. And what we see is that there is a correlation between the egg laid on the plants and the survival of the larva. So, which means the less eggs are laid, the less larva will survive. And this can explain specialization of the beetle for the common ragweed, because sorry, I don't know how to explain well, but the correlation is a performance correlation, and when this appears, the natural selection will tend to select beetles that will stay specialized to the common ragweed. So that was interesting for us, because we don't want to have a host shift, so a change in the host. And so, yeah, strong preference for ragweed, sorry. And finally, we conclude that there is a low risk for sunflower, because more than that, we also did choice tests, and in choice tests, the beetle didn't choose sunflower. And also some colleague from Italy did some forced selection on some sunflower. Nicolas, you want to say something? No? Okay, sorry, I heard something. Some colleagues in Italy, they did some selection. Yes?
You know,
we can't hear you, Nicolas. Okay. Well, I will move on. We wanted to confirm that there was no risk for adaptation of Ophraella Communa on sunflower. And that was done in Italy in a 3-year trial. What they did, that they caged in the field Ophraella Communa with sunflower, and after 3 years, all Ophraella Communa died. So there is no risk of adaptation.
I wanted to bounce on.
Okay, we can't hear you. No. Sorry.
I see that Nicolas' mic is on, but probably the internet connection is not good enough.
Yeah, probably. Sorry,
but
Nicolas.
Maybe you can write in the chat if you have something to bounce on. Nicolas,
yeah.
Maybe it will be better.
That's it for our results. So that's what we used for the application file for ROUSSET, and it was accepted, so it's good. And along with our process, so my master thesis was in 2023, and during my master's thesis, Ophraella Communa was discovered in France just before I started my PhD. So it was quite interesting for us, because we were doing the file for ROUSSET, and then the beetle arrived in the Auvergne-Rhône-Alpes region. So I started my PhD by doing some trials in the field. We had three actions, in the field. We had one that was with the ragweed reporting platform of Fredon, where we used a report from a citizen to localize Ophraella Communa. Another action was the mobilization of ragweed local reference to monitor locally the spread and impact of the beetle in the Auvergne-Rhône-Alpes region. And the third action was of a field study that we conduct to measure the impact on the growth rate of Ophraella Communa in the field. So here you have some picture of the insect in the field, the larva, the adult, and the eggs. So the two first studies that we had was citizen science, and for the local ragweed reference, we had 21 referents that participated, and 14 of them localized Ophraella Communa in the field in their area. And the first detection of the beetle was around June, so quite late compared to the appearance of Ambrosia artemisifolia in the field. That explains a bit why they found quite low foliar damage on Ambrosia artemisifolia. And we don't have big impact yet in the Auvergne-Rhône-Alpes region, unfortunately. But it's just the first step of the colonization, so we hope for a better impact later as the year passes. And for the Ragweed Observatory platform, I will show the map just later. We had 74 reports that I confirm using the picture and using the comments of the referent, because it was people that most of them already knew about the Ophraella Communa appearance because we did a lot of meetings, some of the fields with as we can see in the picture there. And so it allows us to have the detection of Ophraella Communa in 7 departments. For those who knows, Ain, Rhône, Isère, Ardèche, Drôme, Vaucluse, and Gard. Here you have the map. So it's a zoom of the region, of the Auvergne-Rhône-Alpes region. So the first observation in 2023 was around Lyon. It's a blue dot here. And all the yellow ones are from last year, so 2024. You can see that it went quite far in the south, so it's almost 200 kilometers compared to Lyon, from Lyon to Avignon, so it's quite far in 1 year. And we have some missing point here, so we don't know why. Maybe it's because the beetle jumped using maybe public transport, human transport, or maybe we just didn't find it, but we don't know. All the white dots are points where we couldn't find Ophraella Communa. And this is also interesting because we know that it's just arrived in the south, so it's only at some specific localization, so it's interesting. But around Lyon, it's already spreading a lot. And this is a work that we will publish soon, I hope. Work in progress. Here are our field study that we did last year. So we had 100 meters square plots in the Auvergne-Rhône-Alpes region. And we had 10 fields with 20 marked plants per field. And we were doing the sampling every 2 to 3 weeks, from June to September. Here you have the picture of our interns that were measuring the height of the plant. One square that where we had in the beginning of the season where the common ragweed is quite alone in the field because we also looked at the plant competition. So we were looking at the percent coverage of the plants around the ragweed in the quadrat. And to the left, you have the picture of one plant that is completely eaten by a larva. So it's by Ophraella Communa. We were scoring the plant growth, the herbivory damage, like the picture, and also, like I said, the percent of coverage by other plants. And also at the end of the season, we look at the flower because we want to see if there is an impact on the flower production. We don't have much result yet because we want to conduct this study again this year, but here you have the foliar damage during the season that we had. So what we see is that the damage remained quite low before September, so it didn't have enough impact on the plants to reduce the production of flower, unfortunately. But after September, the damage are increasing a lot. Depending on the field, but yes. For example, for Saint-Priest and Vienne, the damage were above 75%, so it's quite high. But most of the plants produce pollen, unfortunately. So we can say it's the very first step of the colonization process because at the beginning of the season, we had no Ophraella beetle at all in our field. They appeared quite late, so they appeared after June. That's why the damage remained low at the beginning of the season, and that's why we will do again the study this year to see if there is an improvement of the damage or not. So study to continue and result to come. So just so you have an example of the damage, to the left you have around 30% foliar damage with a plant that still produced a lot of flower and still have some leaves intact. In the middle, above 90% damage, a plant with almost no leaf left, and that produced one flower, but only one. And to the right, 100% damage, where the larva also eat the flower because you can see, so at the top, you have something looking like a flower, but the larva eat it and then they pupate at the top when they had nothing to eat more. What we are looking for with biological control is damage that are around the 90% and 100% if we can. This was, all the picture was taken at the same field, so you can see that inside the field there is some variation, but yeah, that's why we are waiting for later. At the end of the season, so in October, we started another wintering study where we put some insects in some pockets, like you can see in the field, in the picture. We had 3 subsets, so hay, hemp, and soil that we took from the field at 3 sites that we studied. And it was between October and February, so we get the result last week. So I will be able to show you some results. It's interesting. And we took the adults that are overwintering in the field, so directly on the field, put them on the pocket and wait for 4 months. We were quite afraid of not finding them alive in February, but we have some survival. It's quite interesting. We want to look at the survival rate. Also, I will look at the sex ratio of survivor, but I didn't have the time yet. And also, we want to look if they will reproduce well after the winter. So just the first result, because I didn't have time to do much between last week and this meeting. We had three fields, but we lost one because of wild boar, unfortunately. So we only have Salèse and Saint-Priest, the green and the orange. And what we see is that hemp was not a good substrate for Ophraella Communa to overwinter, most probably because it was quite compact. And the only survival that we had was, like for the picture to the right, they were just standing there in the net, not inside the hemp, so it was not a good way for them to overwinter. But hay and soil was quite good, with a 40% survival and also more than 45% for Saint-Priest. So it's really interesting because it means that almost half of the beetles survived the winter in hay. It's quite good for beetles that just had to wait for winter to pass. And there's some difference between the field for the soil, most probably because we took the soil inside the field, and Salèse was a crop field that was really low in nutrients and activity, microorganism activity probably. And Saint-Priest is more like a meadow, so it can explain also why there are more survival in the soil in Saint-Priest. That's it. I'm waiting for the next results because we have them waiting for them to survive and reproduce. So we will see.
Urgent.
Oops.
Ouais, je regarde, tu sais, là, le truc sur
sorry,
Ophraella.
there's someone we can hear.
Yeah, I turn off his mic. Her mic.
Okay. Thank you.
So thank you, Zoe, for this presentation.
Yeah.
I just see Nicolas is there.
To finish.
Est-ce que vous m'entendez?
Yes.
Oui.
Yes.
Ok, mais en fait, je me fais déconnecter de façon régulière. Donc, la connexion est moisie.
Okay.
Voilà.
You can conclude, Zoe. We are arriving to the end. So
sorry,
yeah.
I was I took some
if
more
you want
time.
to have time for questions.
Yeah. So it was all Outlook. So we will just continue what we did last year and extend our survey, and we will do some lab study. But it's all right. It's finished. Thank you. If there are some questions now.
Thank you very much for this presentation. So if anyone has questions, I have some, so I can introduce this question if you want.
Yes.
Okay. So you have conducted tests with plants in France, which
Yes.
showed very encouraging results.
Mhm.
Are there any documented cases, perhaps from other countries, of Ophraella Communa feeding on plant species other than Ambrosia atriumicifolia? Or
Maybe that's Nicolas that could answer because he knows a lot, but no.
We don't have records of that. So,
Mhm.
and actually, when I started to speak earlier, so I just wanted about the preference-performance relationship that Zoe described.
Yeah.
When you have a positive preference-performance relationship in organisms, like it could be phytophagous, but it could be also natural enemies. Often, what it means is that it will lead to higher specialization to the most preferred host or plant here. Okay, so I think we did not show the preference performance relationship, but in the figure, you know, on the very top right in the regression, in the correlation, then you have one single species, which is common ragweed. All the others, they are clustered way below. So it's kind of an ecological process. And yeah, to reply to Tristan, we have no records of the insect eating something else. Okay, despite being, you know, despite it has been, you know, the subject of inundative mass release in China for past 15 years, you know, on thousands of hectares. So we have no information, no fear.
Okay, I have maybe a second question, and then given that Ophraella Communa was first detected in two separate locations, or not first, but you detected in two separate locations, Lyon and Avignon, about 200 kilometers apart. Is it also possible that there were two independent introductions rather than a single spread from one initial point? And then have you considered conducting genetic analysis to determine whether they originate from the same population?
Yeah, so the date of appearance are quite different. In Avignon, it was in October. In Lyon, it was in June. So we don't know if there could be other introduction. I don't know if Nicolas wants to do genetic analysis. I didn't plan to do that. Maybe some colleagues from INRAe could do that. What we know is that orbiter from Lyon is from Italy, but Nicolas, you want to speak?
Yeah, so we do plan to do a phylogeography analysis. So collecting what we are also very interested is to know all the populations from North America, the native area, China, Italy, and France are related. Because we don't know if the population that actually landed in Italy is from North America or from China. So this we will study that with a colleague. Okay, and regarding the question about the population, I mean the collections, the detections that we had in Lyon and in Avignon, actually there is probably you know, some other points that we did not detect between these two areas, because here there is a big highway and you know, it's a major highway. So I would say that we probably have some, you know, some points that were not detected. Okay, because we have a limited, you know, we have limited capacities. So I would say that it's probably the same genetic background. I mean, the same genetic line. Yeah, but this would be maybe tested, except if they merged next year. I mean, this year when we will do the new, you know, the new scouting.
I also have a question, because ragweed is very common in the Rhône Valley, and so it's a good thing that the beetle was there. But there is ragweed more in the north or so. So I was wondering if the species is able to go across the whole French territory, and how far, probably not, as I understood, but how far Is it supposed to establish how far north, like the Loire Valley maybe, or even more in the north?
That's a good
So
question.
we have a card with prediction. So I don't know if we have it here.
Yeah, it's not there, but maybe if I can find it.
So Zoe, while you are looking for the map, you can share maybe. So the potential distribution of Ambrosia of common ragweed is much bigger. I mean,
Okay.
Than Ophraella Communa. You're right. Ophraella Communa may go to the Loire, to the center of France, but may not go beyond. Okay. It's unclear. And this is the first point. It's about capacity to overwinter and to sustain in that part of France. And also the second aspect is being able to do enough generations to be able to reach the efficient density to prevent pollen release. And you see on the map that is showing Zoe, that maybe Zoe you can explain because it's your map.
It's not my map. It's data from I think Müller, Scherer, or Steffner publication that I took, and I just added my points of observation of Ophraella Communa, and they map by doing a model on the computer to use using climate data and also what they know about Ophraella Communa to map where Ophraella Communa could do 3 to 5 generations, so a lot of generation, and where Ophraella Communa could do only 1 to 2 generation, but still survive in the field, and the areas where Ophraella Communa could be because common ragweed is in the less green part, but will not survive and will not reproduce well. So you can see that it's quite restricted in France, but this is a model, so maybe it will change. We are looking at that. We want to look at that, but that's why we will try some release in Allier region in Auvergne-Rhône-Alpes. So maybe it's around there. I'm not sure, but in a part where Ophraella Communa could not, according to this model, reproduce well. So if we release there, we will see.
Okay.
Maybe, yeah.
But we probably cannot count on it in Belgium.
In
No,
Belgium,
you can because the plan is to have it mass reared and then distributed to the places where it will be needed. Okay. And what is also difficult for us is, for example, there is barely no information about overwintering, habits and capacity. Okay, that's the reason why we do some experiment. So, maybe the insect will be able to stay in a bigger, in a wider distribution. Okay, but this only time will tell.
Okay.
And maybe as a biological control agent, it can still be used in northern regions, maybe.
For inundative release.
Yeah.
Yeah.
Any other questions?
There was a question in the chat, but I replied. Question from Anne-Marie.
Maybe can you repeat it and very quickly and respond, because we are recording right now and people won't have the chat.
So the question was, February survival will be alive in June? Question. And what they will eat before there is ambrosia. So what NICOLAS said and what we know is that we took them earlier in the season that was going to happen in the field, because we wanted to really see if the survival that we have is because they survive winter and not because they survive and not just because of lack of food. So we took them before Ambrosia artemisifolia starts growing and before Ophraella Communa will most probably go out of winter. But in the field, they will go out of winter when Ambrosia artemisifolia start growing. So they will have food when they start emerging. So that's it. And most probably, the ones that survive winter will not be alive in June because they will most probably die soon. They will only reproduce, do one cycle, and they will die soon. So, because when we went in May, it was in May last year, we found only larvae in the field and no adults. But adults were there at some point.
Which is common in the species. It's the same in the native range. It's a normal cycle. Okay, thank you very much. So if there are no more questions, I think we can...
Maybe I will cut the recording. I will just say for the people who are listening it recorded that you can also present your work in future Let's Talk About Ambrosia. So you can send us an email in the description of the video. And thank you very much, Zoe, for your presentation. And we can cut the record, I guess.
Thank you.
Okay. And Nicolas, have a nice day in China.
Thank you.
Okay, thank you.